As they move about their environment, all organisms shed tiny bits of organic material. These trace remnants contain DNA that help conservationists better understand the species makeup of an environment, much like the forensic pathologists on your favorite police procedural. The organic material that animals leave behind is called environmental DNA, or eDNA, and scientists are starting to comprehend how it could revolutionize our understanding of the natural world. To protect the natural world, we must understand it. To understand it, we must study the composition of our ecosystems. Unfortunately, many species are difficult to find and track, especially the ones that are the most threatened. That's where eDNA comes in. When sourcing eDNA, scientists will collect a sample from any number of environments. Water, dirt, ice, and air are the most common. Scientists are even beginning to sample from flowers to detect pollinator presence and parasites to see which animals played host to them. At the collection step, it is very important to use proper technique to avoid contamination, most often caused by the scientist's own DNA. DNA can also be contaminated when it degrades after being exposed to heat and ultraviolet rays. Once the sample is collected, the scientists will either bring it to a lab or use a portable field kit, which are growing in popularity. There, samples will be sequenced for eDNA from any number of sources: skin, scales, hair, feces, mucus, or urine, for example. An important consideration for choosing the appropriate sequencing approach is whether to target certain genes that help to differentiate taxa of interest or sequence more broadly. A scientist may choose to target a specific sequence to search for an individual species or use a shotgun approach to assess biodiversity present. To identify species in the sample, scientists compare eDNA sequences to reference sequences. Thus, successful analysis of eDNA sequence data is highly dependent on having a reference sequence database that contains the species or close relatives in your sample. Once the eDNA is sequenced and the species are identified, scientists can use the resulting data for a variety of conservation purposes. Perhaps the best use of eDNA is to establish a baseline of biodiversity in a given environment. By collecting this data over a long period of time, conservationists can understand how populations change and hopefully diagnose negative shifts in time to address the cause, often anthropogenic in nature. Another use for eDNA is to track and monitor endangered species. These animals are often the most difficult to find evidence of, and having the ability to search an environment for even microscopic presence of a rare or threatened species is crucial. The Wildlife Conservation Society is using this technology to search for evidence of the rarest turtle species in the world, Swinhoe's giant softshell turtle. Additionally, scientists are using eDNA sampling to track invasive species. These animals become invasive often because they are hard to monitor and therefore hard to manage. For example, scientists are sampling ballast water to hunt for elusive invasive fish species. A relative newcomer on the conservation scene, eDNA has both upsides and downsides, pros and cons. Before the advent of eDNA, scientists would have had to trek the field for weeks or months to collect and identify animals in a given environment and set a baseline for biodiversity. eDNA provides a much less work and time-intensive option, requiring only a simple series of minimally invasive samples. This highlights another strength of eDNA. It is far less invasive than other tracking and monitoring methods. Scientists sourcing eDNA can simply take small samples from various sites in an environment, rather than overturning every rock and log in an area and disrupting sensitive habitats. Finally, eDNA is more cost-effective than conventional methods. A small survey near a wind farm in the North Sea involving 3 water samples cost $750 and identified 2/3 of the fish species that were recorded by a 2-year, $191,700 monitoring program. As the technology exists now, eDNA cannot accurately estimate population abundance. While cheaper options are being developed, eDNA sequencing also requires expensive lab equipment and sophisticated bioinformatics skills. Finally, successful analysis of eDNA sequence data is highly dependent on having a reference sequence database that contains the species or close relatives in your sample. However, more often than not, scientists do not actually know what species are in the sample. Large public genetic sequence databases like NCBI GenBank are commonly used, but they are biased towards what researchers contribute to the database. As such, some taxa are better represented than others. The future of eDNA has untapped potential as a citizen science initiative. The sampling method can be so simple that it could be done by anyone. In the UK, an eDNA survey of the endangered great crested newt, collected by members of the public, correctly identified 219 of 239 sites as supporting newts. Given that the volunteers had received no training, the researchers suggest that even minimal training could reduce the false negative rate. Researchers are setting up similar projects with ecotourism companies in the Amazon. Tourists would collect eDNA samples while visiting national parks, thus contributing to long-term biodiversity monitoring. The possibilities to conservation presented by eDNA are boundless, and its role will only continue to grow.